So we've already talked about the diversity that's available within the D-genome ancestor of wheat, Aegilops tauschii. Now the next step is to cross that diversity into bread wheat and evaluate the progeny to look for lines that have useful traits, that have increases in particular adaptation or productivity compared to the wheat parent. So what has been done here is to cross 7 different tauschii accessions, which were considered to have useful traits and which represented a range of diversity, to cross them into the background of a Kansas adapted line. This is work that was done by Eric Olson, who was at Kansas State University, now at Michigan State. But we're growing them here particularly to look at potential drought tolerance traits. I think it's really interesting to look across this field and to see just naked eye differences among the materials here. So most bread wheat and most of the lines in this trial have got awns, these barbed projections from the spiked, but some of them are awnless, like this one here. There are different heights. There are different colors, which I think you can see. Some of them are turning brown and reddish, and some are going to mature into a more golden green type of head. There's differences in maturity. You can tell some of them have already turned yellow and brownish, and some are still green and photosynthesizing. So that kind of diversity is good. That's what we're looking for, to be able to find some that have got traits that can help us out for adaptation to the Great Plains. So this is the side of the field that was fully irrigated all during the season. In an adjoining part of the field, we have had no additional irrigation after the initial germination of the crop last fall. So one of the primary traits we'll be looking at is the difference. How well do these materials yield compared to the yield in the drought stress part of the field? And we hope to find things in the drought stress part that at least come close to their yield here in the well-watered part. But that is one step in the process. The next step would likely be crossing these materials back into an adapted background. But it's a slow process, and it can be painstaking at times, but the hope is that there will be a payoff and that we will find at least a few useful genes from Aegilops tauschii that will benefit our modern wheat cultivars.